Good morning or good afternoon. I would like to welcome you to this brief presentation on current genetics research within ILRI. Firstly, a reminder that global livestock genetic diversity is huge. Globally, there are numerous genetically diverse breeds, including more than 500 breeds each of pig and goat, and 1,000 breeds each of sheep, cattle, and chicken. Many of these breeds are already found in the production systems where we work, and increasingly new breeds are being introduced. In addition, there is genetic variation between individuals within a breed. These variations occur as differences in the DNA sequence from one individual to the next, and there is significant scope for this given the size of livestock genomes. For example, the cattle genome has 3 billion base pairs of DNA, and the chicken genome 1.2 billion base pairs of DNA. So our research focus is to capitalize on this genetic variation. The means by which we can capitalize on the global livestock genetic diversity I just described will be discussed here. Firstly, we aim to match breed types to production systems and environments, taking into account the current and predicted future changes occurring within our target livestock systems. And secondly, we aim to ensure that livestock keepers can access the breed types they desire. On matching breed types to production systems and environments, we are following two main approaches. One is to identify the most appropriate breed type from existing choices. And the other is to create new improved breed types, either by conventional breeding programs, including crossbreeding programs, or via the use of a new technology that allows us to directly edit or change the genome of an individual. On ensuring livestock keepers can access the breed types they desire, we are working on strengthening the delivery systems for improved genetics. In addition, we are working on better systems for on-farm performance monitoring, providing feedback useful for both ongoing research and for the farmers to base their management decisions. This final slide describes the varied science we use in our work. We are performing socioeconomic research, including trade-off assessment of keeping different breed types, and assessment of germplasm production and delivery systems, as well as animal genetic resource use policy. We are also trying to understand system change in the context of animal genetic resource use. On phenomics and recording systems, we're looking at new ways of measuring livestock traits and also developing handheld systems for on-farm monitoring of animal performance. We are looking at methods to map the breed distribution, inclusive of participatory approaches, given that we often don't know which breed types are where. On genomics, we're performing high-end genomics, such as identifying genes controlling important livestock production traits. We're establishing platforms to perform genome editing, and we're also using DNA marker-based approaches to determine the breed type of animals in the absence of farmer-recorded breed type. On animal breeding, we're looking at defining breeding objectives, as well as supporting the implementation of breeding programs. Finally, on reproductive technologies such as artificial insemination and embryo transfer, we're performing adaptive research. I would like to thank you for your attention and very much look forward to further discussion on this.